Well, thank you so much. I'm really excited about the opportunity to share some thoughts with you and get some feedback. So what I'd like to talk about is how, and this goes a little right in line with kind of Alistair's framing, which is that how bioelectricity can help us scale up single cell level physiology states into large scale anatomy and possibly cognition. The thing that motivates a lot of our thinking about this problem is the idea that all of us have crossed this kind of Cartesian cut, moving from so-called just physics, right? So we were all quiescent oocytes at one point, so a collection of chemicals, to one of these remarkable complex creatures, or possibly even something like this, a high-level cognitive intelligence. So this is a single cell. This is a lacrimaria. It has no brain, it has no nervous system, and yet it has very high competency at single cell level agendas. So metabolics, the single cell level behaviors, physiology, all of that, it does perfectly well. So what we really want to understand is this slow and gradual process that goes from simple chemistry and physics into body structure and then cognitive complexity, and how evolution results in hardware that's able to perform that kind of scaling. So the three sort of main points for today I'd like to transmit are these. First of all, to start thinking about all intelligence as collective intelligence, meaning made of parts, where the most interesting question is, how do the competencies of the parts in some way integrate into an emergent, coherent, large-scale individual via this multi-scale competency architecture of nested problem solvers. And we'll talk briefly about that. I want to talk about bioelectricity as a kind of cognitive glue, which basically in the brain, of course, we know it binds, electrical networks bind the activity of individual neurons into the cognition, the plans, the memories, the hopes and dreams of large scale kinds of selves. And specifically focus, I want to tell a couple of stories, because these two are subjects that I've talked about here before. But what I'd like to do is specifically focus on the role of the VATPase as a versatile piece of molecular hardware that is incredibly highly conserved in these kinds of roles, in these processes that I was talking about, and in particular in scaling up from single cell information to a body-wide pattern. So what I mean by nested problem solvers is something like this, which is that biological systems are not only nested dolls structurally, which is kind of obvious, but more importantly, every level here is itself a problem-solving agent in different spaces. So all of these different layers solve different kinds of problems with different degrees and kinds of competencies. And so we generalize this idea of intelligence as some degree of competency in diverse spaces. We follow Wiener and Rosenbluth's sort of scale here of it's a kind of cybernetic approach to looking at what are the different kind of phase transitions in these competencies, right? You can start off with very simple kinds of behaviors and eventually worked up to something very sophisticated. But what we are very good at as humans is recognizing intelligent behavior in three-dimensional space. So medium scale objects moving at medium speeds in 3D space is very easy for us to look at birds and various other creatures doing clever things. But there are actually many other spaces in which biology is also executes very competent navigation. So there are transcriptional spaces of possible gene expression levels. And if we have time, I have some a story I could tell about some amazing navigation of transcriptional space, physiological space, of course, and morphospace. This is what I'll spend most of my time talking about. So morphospace is the space of all possible anatomical configurations for a particular structure and how it is that we get from here to there. So here's an example of anatomical navigation in morphospace. This is a cross section through a human body, and what immediately strikes one is this idea that there's incredible spatial order. So all of these tissues, organs, all these structures have to end up in the right shape, the right size, the right orientation and position relative to each other. And yet we all start life here as a set of embryonic blastomeres, and they all have to create this. So we have to ask ourselves, where does this invariant structure come from? Where is this information? And you know, a lot of people will intuitively say, well, it's in the genome in some sense, but we can read genomes now, and we know what's in the genome. The genome specifies protein sequences and sort of the micro level hardware that every cell gets to have. But the kind of fundamental problem is to ask this as a kind of collective intelligence. How do these cells build these kinds of things? And in particular, not only build the same thing every single time, but actually are able to adjust for all kinds of crazy perturbations, some of which I've talked about in the past. So what we really would like to understand is how do these agents work together to build this kind of a thing? And as engineers, we'd like to know what else can we make them do? And as workers in regenerative medicine, we would like to know, well, if a part of this is missing, how do we convince them to rebuild what exactly goes here? And this idea of viewing morphogenesis as a kind of intelligence really is compatible with very old views, such as this one by William James, which goes like this: intelligence is the ability to reach the same goal by different means. And what I really like about this is that it's substrate independent. It doesn't say anything about brains. It doesn't say anything about specific problem spaces. It focuses on this general invariant that all intelligent behavior in whatever space by whatever kind of agent is the ability, some degree of ability to reach specific goals by different means. So what does that look like in anatomical space? Well, here's an example. So early embryos, for example, mammalian embryos, cannot count on having the right number of cells, because what you can see is that if you cut an early embryo in half, you don't end up with two half bodies, you end up with two perfectly normal monozygotic twins. So this is a system, the early embryo is a system that is not particularly tied to having a specific number of cells. And if that changes, no problem, it can still navigate from this state to that state. It gets even better than that. Not only can't you depend on how many cells you have, but you also can't depend on the cell size or how many copies of your genome you have. So here's an example, one of my favorites. This is a cross-section through a kidney tubule of a salamander. And normally what happens is that each cell is a particular size fitted to the amount of DNA it has. And thus it takes about 8 to 10 cells to work together in cell-to-cell communication to form this tubule. But one trick you can do is you can make early cells in this animal that have multiple copies of the genome. So instead of 2N, you can have 4N, 6N, 8N, and so on, multiple copies of the genetic material. If you do that, the cells get bigger and bigger. As the cells get bigger and bigger, so they adjust to the cell size to the amount of DNA. In fact, what the bigger cells do is they adjust how many of them embark on this construction project. So because they're so large, you don't need that many. You can make the same thing out of fewer cells. So the cells become much larger, the newt stays the same, the tubule stays the same. And the really amazing thing happens here when you make 8N newts, the cells get so gigantic that one single cell bends around itself and still forms the same tubule. What's incredible about that is that this is a completely different molecular mechanism. Here you've got cell-to-cell communication and so on. Here you have some sort of cytoskeletal bending. So in the service of a large-scale anatomical goal, different molecular mechanisms get called up to solve the problem and give you the same exact outcome. So that's very interesting. That's the ability of biology to control cross scales and to call up different kinds of competencies at the lower scale to get you to the same region at the higher scale. So this, of course, underlies things like regeneration, where you can amputate. For example, this is a salamander, the axolotl. They regenerate their legs, their eyes, their jaws, their spinal cords, just amazing. And you can amputate anywhere you want along this axis, and it will regrow exactly what's missing. And then, of course, the most important part of all this is that it knows when to stop. When does it stop? When a correct salamander arm has been produced. So now we know, yes, we can navigate morphospace from here to there, from different starting positions with different hardware. You can still get to where you're going. But now the question is, how do you know where you're going? And here's another example. This one came from my lab from a few years back, where we found out that, the standard kind of story of metamorphosis is that you have a normal tadpole. Here are the eyes, the nostrils, the brain, the mouth. And these tadpoles become normal frogs. In order to do that, all the craniofacial organs have to deform. So the jaws have to come out, the nostrils have to move, the eyes have to move forward. And it used to be thought that this was a hardwired process. Somehow the genetics gives you tissues that move in a particular direction, a particular amount. And because you're starting out in the right positions, you end up in the right positions, and there you are. So we wanted to test the idea that this process actually might have more intelligence than that. And so we created so-called Picasso tadpoles. And these Picasso tadpoles have everything in the wrong place. So, so the eyes are off to the side, the jaws might be on top of the head, like everything is moved around. And this is Laura Vandenberg's work. And so what we found is that these tadpoles actually make perfectly normal frogs by and large, because all of these structures starting off in the wrong position will still will move in unnatural paths. In fact, sometimes they go too far and have to double back until you get a correct frog face, and then they stop. So what we actually have here is a kind of error minimization scheme that the system is able to do. And so what we think about is something like this. This part here is the standard part that's in all of the kind of developmental biology textbooks, which is this sort of feed-forward emergence, this idea that genes regulate each other, they encode some effector proteins that are sticky, or they exert force, or they do various things. And that eventually, by high numbers of cells following the local simple local rules, you get an emergent structure such as this, and that's fine, and that of course does exist, but it's missing a really important component, which is this homeostatic component. The idea that if these structures are deviated from, and that could be injury, mutations, teratogens, parasites, whatever it is, then there are some circuits that kick in both at the level of physics and genetics that try to reduce that error and try to get you back to where you need to be. So the idea of homeostasis, of course, is not anything new in biology, but there are two things here that are kind of different. One is that typical homeostatic loops have a scalar as the set point. So it could be energy level or pH or temperature or something like that. In this case, the set point that you're comparing to has to be a somewhat more complicated data structure, because you're trying to achieve a morphology. You're trying to achieve a particular shape. It's not a number. It can't be described by a single number. So then we have to start thinking about, okay, how do you store more complex geometric representations in cellular parameters? And then the other thing is that it makes a quite strong prediction. It predicts that if this is true, then in contrast to current approaches, which say that if you want to make changes here, let's say repair the anatomy, birth defects or something, you have to act up here. That requires you to try to solve a really difficult inverse problem. Which genes do I change to make a particular anatomical change here? In general terms, that's unsolvable, and it's what's going to limit all of the kind of genome-focused approaches such as CRISPR and genomic editing and so on, because we actually have no idea which genes to tweak for making large-scale changes. But what this kind of homeostatic loop suggests is that there's actually perhaps another approach. If this is true, you should be able to find, decode, and then learn to rewrite the set point. Because if you can do that, the system is now seen to be reprogrammable, meaning that we can change the set point. The cells will build to something different without having to change the hardware, any of this. It's like you could change the temperature on your setting on your thermostat. You don't need to rewire your thermostat in order to do it, because it affords you an interface for changing that set point. And so now the question is, does biology afford us such an interface? And so this is what we've been looking for and working on for a long time. One of the really interesting things here is that this kind of any kind of a homeostatic process like this is a very basic, simple goal-seeking system. That homeostatic set point is the goal. And